if this career doesn't pan out, I'm going to be a weatherman. I just like watching the weather.
I'm pretty sure it's already panned out.
Yeah, the radar is kind of cool. So you know you get the radar on your phone? Isn't that's like the best thing that can happen with your phone, is being able to watch the precipitation roll into town. It's pretty awesome. Okay, what kind of dog do you find in a bakery? What kind of dog do you find in a bakery? What was that? Hot dog. In a bakery? Mostly like breads. But that's a good guess. I do like that. Purebred. Nicely done. Maybe a purebred hot dog. We'll combo those. What is a frog's favorite kind of flower? What is a frog's favorite kind of flower?
A lily.
A crocus. A crocus. You guys not know what a crocus flower is? Oh my goodness.
No.
Didn't you take Bio 182?
Yeah. We didn't talk about that
one. Croci in there.
No.
No? Isn't croci the plural of crocuses?
Maybe, probably.
I don't know, grammar was never my thing. Scientist, so. Thank goodness for the squiggly red line under the word not spelled correctly.
Yeah.
That was a brilliant, that should have been a Nobel Prize right there. Okay, you just right-click it and then it turns into the right word. I mean, I remember my grammar teacher told me that I'd never amount to anything for two reasons. One, I talked all the time in class, okay? And number two, because I couldn't spell very well. But look, she was so wrong. Now I get paid to talk in class. And I don't even have to spell anything because it's always underlined in red squiggly. So she was wrong times two, so there. And she only called me Robert. She wouldn't call me by any other name because that's what my legal name was. Did anybody have a... Maxwell, did you have a teacher like that? That only called you Maxwell? Wouldn't even call you Max. Yeah, see? Yeah, I think same generation right there, man. Not you and I mean our teachers. I wasn't trying to bring you down to that level. Sorry. My bad. Okay. All right, so our bonus 3 is turned in. Exam 4 is upon us. And my mic just went out. Oh, it's back? Oh, we're doing that again. Okay. We'll see how it goes. I might need a backup mic. So this is where we left off. And right when we ended, we actually ended a little bit early. It was about 60 seconds early. Whoa. Yeah, it was. It was about a minute early. You guys don't remember that? Someone's calling BS on that one? I did hear that. We're recording, so I had to edit it with the abbreviation. So it's not as obvious, okay, when you're watching the public recording. Or next time we'll beep, right? So we talked about calcium availability as being really the pivot point for this contraction event. And the mic is off again. Am I doing something? Am I wiggling too much? Or? Sorry, put it on my tab. Okay. Okay. Thank you. Thanks, Patty. Let's give Patty a round of applause.
Woo!
Patty, best. Okay. I must be rough on these. At the end of the semester, if I get a bill for like $1,500 for microphones, I don't know what to do. But anyways, let's talk about calcium. So calcium is critical because I told you last time we were together on Thursday that the calcium is going to go and bind to troponin. And that troponin molecule, in fact, let's back up one slide. That troponin molecule has a binding site for calcium. When calcium binds to it, then it undergoes a conformational change. I'm sure you've heard that terminology before. What in the world does that mean? It means a physical change in shape. So imagine Imagine if you're holding a rope, okay, and someone hands you a giant exercise ball, like this big, right? You're probably going to have to put the rope down and grab the exercise ball. So you just went through a conformational change to hold on to calcium, and you had to lose your old shape in order to do that. That's what a conformational change means. So calcium binds, and now that molecule has a different shape. When it has a different shape, because it's physically attached to tropomyosin, so now let's pretend that that rope is attached to my belt loop via a carabiner. Okay? And when I go to grab the exercise ball, it's heavy. And so I grab the exercise ball and I drop down almost to one knee maybe. Well, that tugs on the rope, and so the rope moves as I move. So that's exactly what happens here. Now, this tropomyosin, or the rope in our analogy, is actually physically blocking a binding site on the actin molecule. And that binding site on the actin molecule has a super high affinity for the myosin head. So the myosin head is going to be nearby. It's going to find the binding site on the actin molecule, and it's going to bind. And all of that is regulated by calcium. And so we said that our two regulatory proteins are tropomyosin and troponin because they're connected to each other. But the calcium is binding to the troponin. So that's where we ended, but we're going to keep with the same exact story. We're just going to fill in more of the details. So we're going to talk about it again. We ended at the very end of lecture at 3:24 in the afternoon, last Thursday. We ended with, how do you reverse the whole process? And you appropriately answered, what do? You resequester, although you didn't say that word, but you bring calcium out of the equation. And so the fancy word is you resequester, or you recapture calcium into a storage facility. And that storage facility is this sarcoplasmic reticulum, abbreviated SR instead of VS. Okay? SR. You could say that at Thanksgiving, right? That'd be funny. No, it wouldn't, because Grandma and Grandpa would be there. Never mind. Don't cuss in front of Grandma and Grandpa. Although SR is not a swear word. But my kids think it is sometimes. When I say these words at the dinner table, they're like, Dad, language. It's like, it's actually just a science word. It's not a bad word. Oh, okay. So the sarcoplasmic reticulum is a storehouse for calcium. And the very edge of the sarcoplasmic reticulum is called the end tank of the sarcoplasmic reticulum. Well, in English it translates from the Latin to terminal cisternae, right? So terminal cisternae is the Latin terminology. The English translation is the end tank. Okay, so as we transition into today's lecture, that was the background of where we left off. All of this stuff that's happening is happening right down here. So you can see the thick filament with the myosin globular heads. Do you see that purple, thicker filament right here? And look at how in close proximity it is to the thin filament. And if you look carefully, that thin filament looks mostly orange because a large component of it is actually actin. And the actin houses the binding site for the myosin head. But without calcium present, the binding site for myosin on the actin molecule is blocked. It's blocked by tropomyosin. And then you can also see that here's the elastic filament in green, which is not labeled. You could draw an arrow to it and label it if you wanted to be complete. And what's this structure right here? This Z-zigzag structure. That was a huge hint, by the way. That's the Z-disc, okay? So you know that you're kind of at the end of the sarcomere, because a sarcomere is from Z-disc to Z-disc, so the other one would be somewhere over here. And what this slide is showing is that as these guys interact and slide across each other, they're tugging on something, which is this mechanism within inside the cell. And unless it's anchored outside of the cell, then you don't translate the force from the inside of the muscle cell to outside of the muscle cell. So in order to translate the force that's happening inside the muscle cell as it contracts, is you actually have to have these linking proteins, or what we call accessory proteins. So there's at least 7 that we know of, and we say at least because there's probably more. And they anchor this myofilament structure, this architecture inside the cell. They connect it or link it to the outside of the cell. So when they contract on the inside, it translates the force to the outside. Okay? So the analogy would be, if you have a boat anchor, right, and you don't put the anchor at the bottom of the lake or on the beach, right, on land, and bury it, when you tug on the anchor, nothing happens. It has to be linked or connected to something outside of the boat in order for, when you pull on the rope attached to the anchor, for the boat to actually move closer to shore or move closer to where you want it to be. So you have to anchor or link this thing to something outside. Now, the one that we're going to focus heavily on as we translate clinically is a linking protein known as dystrophin. So dystrophin is this red-colored protein here that connects directly to the thin filament. And then there's other linking proteins that go through the phospholipid bilayer. Now, dystrophin or dystrophin links this entire mechanism to the outside, like we just talked about. So it transfers all of that force outside. Well, it uses other ones as well, but the one reason that we're focusing on this is because this is the disabling disease cause known as muscular dystrophy, is when you have a defect in the dystrophin molecule. So in muscular dystrophy, there's a whole group of muscular dystrophy. We're going to focus on one particular version of it. It happens to be one of the most common forms, and this is Duchenne's muscular dystrophy. It's a sex-linked recessive trait, so it primarily affects boys. So it's 1 in 3,600 live births of boys that actually suffer from muscular dystrophy. It is a disease that can affect females in very rare cases. It's usually diagnosed between the ages of 2 and 10. And the mutation in the gene that codes for dystrophin or dystrophin is what's missing in these patients. And so, because the actin is not linked to the sarcolemma or the plasma membrane, that's another word for plasma membrane, then the cell membrane gets damaged during contraction because it's not connected, so as it contracts, it's kind of aggressively causing trauma to the membrane. Well, that heals over time, and it leaves a scar tissue. And eventually, repetitive scarring causes necrosis of that tissue. The average life expectancy is about 25 years of age in these patients. And it's usually respiratory and cardiac muscle that fatigues or wears out. And so that's what takes the patient. So understanding these mechanisms of how these things work are going to help you explain or understand the type of patients that you're likely going to be dealing with or helping in the future. And so it's kind of nice to be able to put the puzzle together at some level. Okay, so let's move on to a new topic, and let's look at this relationship between the nerve and the muscle. And we call that the neuromuscular junction, NMJ. The relationship between the nerve and the muscle is stated right here. Skeletal muscle isn't going to contract until it's stimulated by a nerve. And I said that even in the very beginning of the class when I was talking about the flow of topics. And that's why I put nerve physiology prior to muscle physiology. Even though your textbook chapters seem like they're out of sequence. It's not that I, just like breaking the rules, but I just don't think that it makes sense to have muscle contraction happening sequentially in the semester until we learn about how you get an action potential to the muscle. Okay, because cardiac muscle is autorhythmic. if you remember that. You're going to learn more about cardiac muscle next semester. So cardiac muscle doesn't need to be innervated. It'll produce a contraction all by itself. Now, you can innervate cardiac muscle, and you can slow it down or speed it up, right? And that's what happens when you exercise or when you go to sleep. But if you transplant a heart from one patient to another, you don't reconnect the nerves. And so the heart just will beat at its basal rate, which is about 100 beats a minute. And in order to slow it down, because it's kind of hard to fall asleep, so the patients will complain, I have like a racing heart when I'm trying to go to bed. So you put a pacemaker in and you actually regulate it down. So skeletal muscle is different than cardiac muscle. It's not autorhythmic. It won't beat on its own. It won't contract on its own. So you actually have to have the nerve going to it. Now, if you damage the connection, the nerve connection, if it's poisoned or if it's damaged or severed, then you have some sort of paralysis that ensues. Denervation, atrophy, is shrinkage of the muscle or loss of size of the muscle when the connection is no longer there. So in patients that have spinal cord injury, the distal extremities, whether it's quadriplegia or paraplegia, so if it's quadriplegia, it's all four limbs. If it's only quadriplegia, then maybe just the legs, then you're going to get atrophy of those muscles because they're no longer innervated by a nerve signal. Okay? They're not being used, that's true, but simply cutting the nerve will lead to atrophy independent of lack of use. So if we look at how this organization exists between the nerve and the muscle, we need to introduce this topic known as the motor neuron and the motor unit. And the motor neuron and the motor unit are defined in different ways. So somatic motor neurons, these are nerve cells who have their cell bodies in the brainstem and the spinal cord, and they serve skeletal muscles. The somatic motor fibers are the axons that go to the actual muscle itself. And the nerve fiber branches out to a number of muscle fibers. And so what you're going to see is one nerve fiber is going to control a number of muscle fibers. Right? So the nerve fiber is sort of the commander. And the army that it has to influence or to lead is the number of muscle fibers that it actually is controlling. So the muscle fiber itself is only supplied by a single motor neuron itself, but there might be multiple fibers. So let's take a look at how this architecture would play out. So here's a spinal cord slice, right? This is a transverse slice. You can see motor neuron number 1 in blue and motor neuron number 2 in red. Now, the blue motor neuron, which is conveniently colored blue for this example, is innervating onto only the blue fibers of muscle. And the red is conveniently only innervating onto the red ones. And you have these two different motor neurons that go to a distinct population of muscle fibers. And in any muscle belly, any individual muscle, let's say the biceps brachii or your triceps or the quadricep muscle group or the hamstrings, you're going to have multiple motor neurons going to those muscle groups. But if you're going to just walk up here, or if you're going to lift this laser pointer, you don't need to recruit all the reds and blues. You might only need to recruit the blues to do this motion. But if I picked up a statistics, that's heavy. A statistics textbook, right, I might need more motor neurons recruited in order to do the same motion, but there's actually a greater force. Okay? So that gives you an idea of, with the same muscle or muscle groups, you have the ability to recruit muscle fibers appropriately. And what I mean by that is, think about it. If every time you did a motion, it was with the same force, that would be problematic, right? If you were trying to crack an egg versus if you're trying to open a jar of mayonnaise, or if you're trying to write with a pencil, or tie your shoe, or pick up a large heavy box off it, you need the ability to recruit a different number of muscle fibers depending upon the appropriateness of the force that you want to output. Make sense? And everybody's had this situation where the sensory feedback gives you information to the muscle saying, okay, this is going to be heavy. You go up to a box and you're like, okay, this is going to be, right? You're like, whoa, that was way lighter than I was expecting. So you just tricked your body into recruiting more muscle fibers than was necessary for the task, because the visual feedback told your body, recruit motor neuron 1 through 10, because that's going to be heavy. Right? I mean, these are things that are happening reflexively as you're seeing the sensory input. Okay? All right. So the motor unit is defined as a nerve fiber and all the muscle fibers that it innervates. And the muscle fibers from that one motor unit are going to go throughout the entire muscle. They're going to contract at the same time, or in unison. And they produce a contraction over a wide area. It says weak contraction. What does that mean? Well, the individual muscle fiber isn't producing a large force, but if you send it to 1,000 muscle fibers, and they're all contracting together, right? That's, kind of like the tug-of-war analogy, where you got a team of 12 individuals tugging on the rope at the same time. You're generating a large force production versus just one individual. The ability to sustain long-term contraction as you recruit motor units, taking turns. So that happens right now as our postural muscles are being exercised. You're not thinking about it, but they're being exercised right now. And as a set of postural muscles is starting to fatigue, you recruit a new motor neuron to actually a new motor unit with the motor neuron that innervates the fibers to keep you sitting up straight in your chair. The effective contraction requires really only the contraction of several motor units at one time. It doesn't necessarily need all of them, but it needs several in order to perform the activity. So if we look at the average architecture, the average motor unit in our body has 200 muscle fibers for each motor unit. About 200. So you have a neuron going from one nerve fiber to 200 muscle fibers. Now you can see two different variations. If you want a fine degree of control, like at your hands and fingers, and even at the level of the eye muscles, right? To be able to quickly move the eyeball around, and then even to help focus. Okay, the muscles that control the shape of the lens. These are smaller motor units, where you've got 3 to 6 muscle fibers per neuron. So you have one neuron going to only 3 to 6. It's a 1 to 6 ratio, right? Now, a large motor unit is going to be more strength. And so if you think about large muscles, like the quadriceps, the hamstrings, the biceps, the triceps, the gastroc, these are much larger muscles than hand muscles. And so in these situations, you've got a large motor unit that has about 1,000 muscle fibers per neuron. So it's a 1 to 1,000. And that's more forceful production. Okay? It's not necessarily able to have delicate or fine motor control, but it's very powerful. So this class is about 1 to 400. So imagine 1 to 1,000 is more than 2x of you and still just one neuron. Okay? So, but the amount of force production that you can create is extremely high. All right, so now we need to transition into the synapse, where all this takes place, the neuromuscular junction. And that's the interface between the neuron and the muscle. And in our previous unit, we talked about a neuron going to a neuron. So here it's a neuron going to a muscle. And the neuromuscular junction is where the target cell, instead of a neuron, is actually a muscle. So neuromuscular junction. In between we actually have this gap or this synapse, and the nerve fiber is going to stimulate the muscle at numerous locations across the synapse. And the reason for that is you want to make sure that the signal gets sent. You don't want to risk the scenario where you say contract and the signal wasn't actually heard. Okay, so you're going to send that signal across in multiple locations. So we'll look at some examples here in a second. The picture that I'm going to show here is a high magnification view, but it's actually a really good summary slide. Because this stuff down here, does this look familiar at the bottom of the picture?
Yes.
What is all this?
Sarcomere.
What's from exactly right here to right here? That's a sarcomere. Very nicely done. This is also considered the functional unit or the contractile unit of muscle, right? Inside the sarcomere, you see the organization, which is the thick filament, and then you see the thin filament. You can also maybe barely appreciate some green right there, which is the elastic filament. It doesn't show up as well here because it's like a yellowish, creamy background, and there's green on top of it. But here is the membrane surface. You see this? Now the membrane surface kind of dives in. It has these invaginations here. But this is actually the sarcolemma, or the plasma membrane. So we're clearly inside the cell, and if it wasn't obvious enough to you that we're inside the cell because of the myofilaments, you see mitochondria, you see nuclei, right? Someone asked last time, is skeletal muscle multinucleated? Yeah, you can see there's 2 nuclei in this particular cell. So you know that you have cellular organelles, you know you're inside the cell. Here's the nerve, and some things that should be familiar to us is here is the nerve axon coming down, and it's covered by this whitish coating, which is what? The myelin. And the Schwann cell is located within the myelin, embedded within the myelin, because the myelin, as it's produced, it wraps around the axon, kind of like we gave those examples of like pigs in a blanket, or making cinnamon rolls where you kind of have to roll them out. And so you get like these wrappings that are layering around the axon as an insulator. So the motor nerve fiber is underneath there. This arrow or this line should be pointing to, I think it really should be pointing here. Because it looks like it's almost pointing to the myelin. It's like, well, what isn't that the same thing as this? Well, this is kind of pointing to the outer covering, and this is actually pointing to really the nerve fiber itself. And then the end button, or the knob, kind of bulges out right here. And if you see within here, this is a cell. So this is one cell here, this is a second cell down here. That's why there's a mitochondria drawn right there. The artist is trying to make it clear that you have cell number 1 up top and cell number 2 down below. And there's a clear gap, right? And that gap is our synaptic cleft. So we're going to take our electrical signal, we're going to transfer it to a chemical signal, and then it's going to bind over here, the chemical, to its receptor, and then that's going to initiate another electrical signal to happen in cell number 2. And this is all at the neuromuscular junction. So the synaptic knob is that swollen end of the nerve fibers. It kind of bulges out. So that's this part right here. See how it's kind of bulging out, fatter than just the nerve fiber itself. The synaptic cleft is the gap between the synaptic knob and the muscle sarcolemma, or the muscle membrane. And we have the Schwann cell that envelops and isolates the neuromuscular junction. So if you back up here, you can see this Schwann cell is located at the neuromuscular junction as well. See how they drew the line down here? So it's laying down membrane here, surrounding this. It's like saran wrap around the junction, or the synapse. Now, why do you think you would have that there? Why would you have like a wrapping or an encapsulated synapse? Yeah, exactly. So if you're going to release valuable neurotransmitter known as acetylcholine in this example, and it's going to go right here into the cleft, You don't want it just to like, just go off. Like, oh, I don't feel like really contracting right now. So you contain it. You keep it here. It improves the efficiency. Improves the efficiency. So that envelops it and keeps it from dissipating out. Now, it's still going to dissipate out, but not as fast of a rate. Okay? So it's more likely that you can actually preserve your signal. Now, the synaptic vesicles at that cleft go through exocytosis. They fuse and they dump their contents. We've seen that before. We have greater than 50 million acetylcholine receptors in the body. So it's very prevalent, right? So why it's the one we study most often in this class, in this Bio 201 class, is because of their prevalence. Now these junctional folds. Look at these junctional folds. So this isn't just a straight membrane. We went through the energy to actually have these invaginations to make more membrane. Now, why would we do that? Increase surface area. So you're going to send out a signal, and now you have a greater surface area to insert acetylcholine receptors. You see how the receptors, kind of these bluish rectangles, they're down into the cleft as well. So you're maximizing your opportunity to bind the acetylcholine. Now, if acetylcholine is released over here, over here, over here, and over here, you're going to be able to bind it at all those different locations. And so the chances of you preserving the signal of the action potential are much greater, because you're going to detect that neurotransmitter coming across that synapse or that gap by having a larger surface area with much more neurotransmitter. Now, an interesting fact of paralysis disease, known as myasthenia gravis, is a lack of these acetylcholine receptors. Myasthenia gravis is a disease that translates to muscle weakness. So in these patients, this is a genetic disorder. They actually have difficulty performing a contraction. And it's not because they can't send the signal, it's because the receptor on the other side is in low population. Okay, so neurotransmitters on the other side. The last little detail that we need to talk about before we get into a couple of images that characterize what this space looks like. And then we're going to move into some toxins and some poisons that cause problems. And these are kind of interesting because I think you've heard probably of some of them. So the basal lamina is this thin layer of collagen and glycoprotein that surrounds that Schwann cell. And it's that extension of the myelin that's called the basal lamina. This basal lamina keeps the content within the cleft. And so what you have to do now, when you want to stop a contraction, is you have to release an enzyme to break down acetylcholine into its constitutive parts. And it breaks it down to an acetate and a choline that get reabsorbed, and then you can put them back together to make new neurotransmitter. So it's also very efficient. Recycling of the neurotransmitter. So back to our cleft. So here, this part of the product that the Schwann cell manufactures is called the basal lamina. And it surrounds, it's that saran wrap that we were talking about that goes around the entire neuromuscular junction. Now, if we look at some high-powered images, right? This is light microscopy, but pretty dialed in. This is 100 microns. You're seeing the motor nerve fiber. This is not a drawing. This is actually a real specimen. All right? This is a drawing, in case anybody's curious. But this is real. Okay? So this is a motor nerve fiber coming down, and this is the end of that termination point, what we call the motor end plate. We refer to it as the motor end plate because it's a motor neuron, and when it innervates onto a terminal target, it's an electrical term to call it a plate. In fact, we're going to call it an end plate potential here in a minute. And the end plate potential gives you a clue that you're talking about an electrical signal going to that target. So, and then you can see that these are the muscle fibers that are coming down like so. The neuromuscular junction is beneath this, right? So if we look at some additional high magnification views, electron microscopy, and we use that same pseudo-colored blue. Here we're highlighting our skeletal muscle fiber. Here is the axon of the motor nerve. This one's kind of cool because you can appreciate how the axon comes down, and before it turns into the end plate, you're seeing what it would normally look like, and then here it's pseudo-colored with the end plate. If we look at some additional high magnification views from electron microscopy, we're looking at our neuromuscular junction. So the primary synaptic cleft is just a gap itself. The secondary synaptic cleft are where those folds, those junctional folds, or those invaginations come in order to increase surface area, and that's what you're seeing pseudo-colored blue down here. So if you took this blue and added this blue to it, you'd be getting this entire membrane with all of the folds. Does that make sense? And then over here, you're seeing the mitochondria that are located at the synaptic terminal. And here are all the synaptic vesicles that contain neurotransmitter, acetylcholine. Again, all of them being highlighted in the pseudo-colored blue. So let's talk about some of the toxins and where things actually start going wrong. If we look at, oh, sorry, I got one more. Neuromuscular, here's our skeletal muscle fiber. This is actually a nice picture because this is the junctional fold right here, right? In fact, it's highlighted down here. But you can kind of see and appreciate, this is the terminal end of the neuron. This is the muscle, right? Over here, these are all mitochondria. They're located within the axon itself. And here are the smaller spheres, or the vesicles. So you can see the vesicles are much smaller than the mitochondria. Here's our secondary folds, or our junctional folds. And here's our primary synaptic cleft. Okay, side view, yeah.
So is the neuromuscular junction actually inside the end plate?
So the terminal end plate is where the electrical signal is going to go. So the electrical signal is going to go to the muscle. So the motor end plate is where the electrical signal will ultimately end up. So the neuromuscular junction and the motor end plate, this would be considered the motor end plate, the skeletal muscle fiber. Okay? That's where you want the signal to go. Because the end game is to send the electrical signal to the muscle to tell the muscle to contract. Okay? It's a good question. So these pictures, I think, are very helpful because it gives you an idea of what things look like. I haven't seen a lot of books that have these types of pictures. Okay, so let's talk about paralysis. Now, we've got some toxins that can interfere with the synaptic function. And some of them are pesticides. And they contain a cholinesterase inhibitor. And so they bind to acetylcholine esterase, and they prevent it from degrading acetylcholine. So now acetylcholine is going to continue to stimulate the receptor. And so what you have is what we call spastic paralysis, meaning there's a paralysis mode because you can't stop it, but it's spastic paralysis because it's in a continual state of contraction. And so with those pesticides, they're going to cause suffocation. That's how they work. They kill the organism by suffocating the organism. Another example, tetanus, or what's known as lockjaw. This is another form of spastic paralysis caused by Clostridium tetani. And in this situation, the spinal cord typically has glycine that's being produced, and this actually stops motor neurons from having these sort of tremors. these unwanted motions that take place. So the tetanus toxin blocks the glycine release and causes spastic paralysis of the muscle. So this is the spastic paralysis example. Now, the next set of examples are what we call flaccid paralysis. Right? So flaccid paralysis is a state where the muscles are limp and cannot contract. And so one example for flaccid paralysis is curare. So this is a poison that's found and harvested out of South America. And it's historically been used to coat these darts for these hunters. And it blocks the acetylcholine receptor because it competes with acetylcholine for the same binding site. And then acetylcholine can't find it, and so you don't contract the muscle. And so now the organism is thinking, run away, run away, run away, but it's laying on the ground and can't move. Okay? That organism could actually be you, for example. Okay? So here's a picture of the plant and how they're being used. Now, the botulism, this is an example that we've already talked about this semester. So I like to bring this one back because it is review material, but it helps put it into context. So this Clostridium botulinum, this is a neuromuscular toxin. Again, this cleaves the SNAP25 protein, which is responsible for helping acetylcholine vesicles dock and dump out their content. And so if you can't release acetylcholine, then you also won't contract the muscle on the other side. And so this is flaccid paralysis operating in a different way. Instead of competing with acetylcholine up top, here you're actually preventing it from being released. All right. At the end of this lecture, we're now getting back to the sliding filament concept that I introduced. Now this concept is being rewritten. The model is being rewritten. It's actually being rewritten by a faculty member here at NAU. And so Dr. Nishikawa is working specifically on the titin protein and has proposed some new models of this theory that alter it a little bit. So probably in about 10 years, okay, in about 10 years, the textbooks are going to start to show a slightly different model than what we're showing today. And remember, I told you that's how this works, right? So the primary literature is peer-reviewed publications, like what Dr. Kisa Nishikawa's lab is writing right now. And then from peer-reviewed publications, we write review papers where we make summaries about this field and all these publications. And then from the review papers, we write textbooks. And so by the time a textbook comes out, you know, the original findings are pretty old. It's not that they're inaccurate. It's just they might not be state-of-the-art complete. So it's not that we're teaching you wrong information. We're teaching you information that might have other aspects to the story. And so the story that you'll probably learn about in the next 5 to 10 years is going to be a bigger role of titin. But right now, we really emphasize the role that the thick and the thin filament play. And the role that titin plays, what we say is that the elastic filament helps with the elastic recoil, the relaxation phase, which is not incorrect, but there's a lot more to that story that we don't spend a lot of energy on quite yet. Okay? So, the sliding filament theory, this was a theory that's ready for revamping. It was originally proposed in 1954. Okay? TV was black and white. Okay? It used a tube. Okay? There was no such thing as a flat television. And that's when we originally learned about this sliding filament model. It was developed by Andrew Huxley, and Hugh Huxley, and Gene Hansen, and Rolf Nietengard. Okay? And they were all working on this in 2 different laboratories, and they sort of collaborated and published the sliding filament theory. And it's not that we think that the theory is wrong, we think that there's other things that are happening within it, which actually does make sense. But the basic principle here is that the thick and the thin filament are going to slide across each other. Okay? Now, the way that the Nishikawa lab is thinking about things these days and the new models that they're writing is there's a bigger role of titin, the elastic filament, being wound within this model versus just being stationary or sitting there. Okay? But you can see this is a relaxed state. If you look at the Z line to the Z line, and you can see that the scale bar is the same, 595 nanometers, that this is the sarcomere relaxed, and here it's contracted down. Okay? So it obviously, these original observations were interesting because they started noticing that in a relaxed state, the sarcomere was bigger. In a contracted state, the sarcomere was smaller. But the relative size of the filaments didn't change, so it led to the idea of, well, they must be sliding across each other. Kind of like that monocular example, the pirate monocular, how it kind of collapses down. So if we further investigate this, we can see some electron microscopy on the right, and we can see some drawings or some schematics on the left. Now, the Z discs are pretty obvious. So if you look at a relaxed muscle here, you've got the largest length of the sarcomere that you can actually observe. And that corroborates with the drawing over here. Now, as you start contracting, you start scooting the Z disc closer and closer, and what we understand is happening is the thick and the thin filaments are starting to slide across each other, but they're not changing in length. When it's fully contracted, and in a fully contracted state, you can see there's a lot of bunching up that's taking place, and you can only contract to a defined physical size or length, because then the structures start getting in the way. Make sense? And you can appreciate here in a fully contracted state, the Z discs are also in this format. Okay, so now we know the architecture. If we zoom down into the muscle fiber, we talked about how the nerve innervates. We send an electrical signal. It crosses over this gap with a neurotransmitter. It binds the other side. Something magic happens, and the electrical signal continues. And then magic happens again, and calcium binds to troponin, and then it slides. Right? So there's a few pieces of magic that we still have to talk about. You guys with me? Okay. So, the action potential. Magic number one. The action potential needs to move from the nerve ultimately to the muscle fiber. So we've got depolarization that we've talked about before. And we have sodium and potassium, just like what we talked about before. And then we have to propagate that signal. So we have what we call voltage-sensitive calcium gates in the muscle. And they're sensitive to voltage, so when the electrical signal gets to them, they open, just like what we saw with the action potential. We had voltage-sensitive sodium gates, and we had voltage-sensitive potassium gates that were responsible for repolarization. So we have another type of voltage-sensitive gate, but it's at the level of the muscle. Why do we care that it's calcium? Because you've already told me that calcium is necessary to become available inside the cell to bind to troponin. And so that's how you couple the electrical signal to the contraction event. Okay? And then, of course, we have to repolarize. So let's take a look at these pictures in higher mag view. Okay? So the nerves tell the muscles to contract using action potentials. And we've already studied action potentials. So we're going to use them again in this particular unit as we wrap it up to describe what we refer to as excitation-contraction coupling. Excitation-contraction coupling is a large section within your chapter. It would be a smart idea to read it. But if you read it once already, it was probably like, well, I'm not really following. Okay? So then we talk about it. Okay? Maybe you go to SI. You draw this out a few times. Then go read it again and see if it's starting to come to life now. after you've got a story and an understanding of how it works. Okay? So don't ignore the section in the text. It's actually really good. It's a really great way to study this, is to actually read the text. And I'm not advocating you got to read the entire textbook from cover to cover. I'm saying when you study, look at what we're going to cover in lecture and go read those segments before you come to class. And then when you read those segments and you come to class, you're going to be familiar with this. Okay, and then afterwards, read it again maybe to see if you understand this concept. Excitation, contraction, coupling is a very, very important concept in this particular unit. Okay, so we know that we've got the electrical signal coming down. I need to back up. Sorry, I skipped all this. So we have the electrical signal coming down, and the electrical signal arrives at this button. This stimulates acetylcholine to fuse with the membrane. It dumps out acetylcholine into the gap. Acetylcholine binds with its receptor, right? So this little box is blown up, showing a larger view of the acetylcholine receptor. Now here's still a larger view of the acetylcholine receptor. That's this one. This is those little purple rectangles that we were looking at. And you can see that there's two binding sites for acetylcholine. And right now, right down the center, the channel is closed. Well, when acetylcholine binds, it opens the channel. And look at this. Same color as before. Our orange-colored sodium rushes in. And we also have some potassium that leaks out because it's not a specific channel. Okay? But sodium comes in faster than potassium is going to go out, like what we saw before. And this is inside of a cell, right? This is a muscle cell. So the muscle cell, this is where the acetylcholine receptor is. It's allowing sodium. The sodium influx now triggers an electrical potential within the muscle membrane. Okay? So you just took the electrical signal from the nerve, using a neurotransmitter, getting across the gap, or the cleft, binding to the acetylcholine receptor here, opening a channel, allow sodium to flow out. Flow in. Sodium to flow in. That causes a depolarization event. Right? Same words as last time. Now, if we have a depolarization event, here's the sodium voltage sensitive channels that open, right? And our potassium voltage sensitive channels that open. This allows for depolarization. This allows for repolarization, just like what we saw before. So these don't happen at the same time. This one happens first, and then it repolarizes next, just like what we saw with the nerve. Right? So there's an action potential with a depolarization side and a repolarization side. You guys following? Okay. So now we have a voltage signal coming down the membrane. And as it comes down the membrane, it's going to run into a pothole. You guys remember what that pothole was called?
Synaptic.
It was a hole in the membrane that dove deep into the membrane. T-tubule. Who said that? Nicely done. T-tubule. So here's our T-tubule. So this arrow sequence is showing the electrical signal coming across the membrane, diving into the T-tubule. Why is it important to go into the T-tubule? Because on either side of the T-tubule is located the terminal cisternae of the sarcoplasmic reticulum. So this picture is actually just referring to this as the SR. This picture now says it's the terminal cisternae of the SR. You guys with me? So they're not really incorrect. It would have been nice if they would have consistently labeled this so it doesn't confuse you. But I'm telling you right now, so this is the terminal cisternae of the sarcoplasmic reticulum, just like it's labeled right here. It should be the same label. I don't know why they did that. Okay. So now you've got the electrical signal coming down, and it stimulates a calcium voltage-gated channel to open, and that dumps calcium out of the sarcoplasmic reticulum into the cytoplasm of the muscle cell. And now, calcium, these little white spheres, can go bind to troponin, and we're going to complete this part of the story that we've already learned. When it binds to troponin, it causes a conformational change, it pulls on tropomyosin, right? That moves tropomyosin out of the way. And now can you see right here, there's this little hole. That's the binding site for the myosin head. Oh, there it is. It's ready to go. So as soon as the binding site is available, the myosin head has a tremendous affinity for it, and it's going to bind. And we call that a cross-bridge formation. Okay? So this is excitation-contraction coupling, and the pivot, the most important component of excitation-contraction coupling, is moving calcium. You have to move calcium in order to couple the electrical signal to the contraction signal. Right? Okay. So now we've got calcium available, and we're going to cycle through these pictures. We're cycling through these pictures because this is how the filaments slide. So, as we cycle through these pictures, let's start up top here. So, this is a uncocked position, like a hammer on a gun, and this is a cocked position. You see how there's the arrow that goes from this position to that position? So this myosin head has bound to it an adenosine triphosphate molecule. And when it cleaves one of the phosphates, it cocks the head back. That allows that head to be in the right position to find the binding site and create what we call a cross bridge. And of course, this only happens when ATP is present on the head, and when calcium binds to troponin, because the binding site has to be open on the actin molecule. So we go from this uncocked position to this cocked position. We have binding sites open, so we get cross bridge formation. Okay? Then we get what we call the power stroke, which is basically the head of myosin going from this position to this position. And as it fires, it takes the thin filament with it. You with me? So as it does this power stroke, is what we call that move, it moves, and you can see how the three arrows are showing movement going in this direction. And as it moves in this direction, it releases the energy that was still there, the ADP and PI. See how they're flying off? So this power stroke takes energy. Cocking the head takes energy, the power stroke takes energy. And then to release, to let go, you actually have to bind a new ATP molecule, and then it'll let go from the binding site. So this myosin head attaches to what? Attaches to actin on the thin filament. It attaches to actin on the thin filament. And this picture is showing the cocking step, the attachment or cross-bridge formation, the power stroke, and then the release, if you will. Okay? So, one clinical fact that's kind of interesting that we're going to talk about is rigor mortis. Okay? So, rigor mortis is this condition where you get stiffness of the dead body, right, after post-mortem. And a lot of it has to do with ATP availability and calcium. Okay, so hold that thought coming up on here in like another slide. So when the sarcomere wants to relax, well, first off, you got to get rid of the signal. So you get rid of acetylcholine, you reuptake it, or you break it down using acetylcholine esterase. The calcium ion needs to be resequestered or reuptaken, and it goes back into the sarcoplasmic reticulum or back into the terminal cisternae of the sarcoplasmic reticulum. And in order to pump it back in, you have to use active transports. Active transports means what? It's against the gradient, right? Because you have high amount of calcium inside the sarcoplasmic reticulum. You have a low amount inside the cell, and you want to take any in the cell and put it back into the storehouse. So it requires energy to do that in the form of ATP. Calcium binds to a molecule known as calsequestrin. Calsequestrin has a high affinity for calcium, so it kind of like pulls it back in and keeps it there once that channel opens, that pump mechanism opens. ATP is necessary here. So ATP is required to resequester calcium, and it's required to detach the head. Okay? So if you don't have ATP, you keep calcium available, the binding sites are open and the myosin sticks. And if you don't have ATP, you don't get the myosin head letting go from the active molecule, and that's rigor mortis. Okay? Rigor mortis is cross-bridge formation post-mortem in the patient, and it'll stay that way until the proteins start to deteriorate. So rigor mortis is a hardening and stiffening of the muscles. It starts about 3 to 4 hours after death. The sarcoplasmic reticulum, the bag of calcium, starts to degrade early, and that dumps a lot of extra calcium into the cell. The sarcolemma also deteriorates, and any calcium outside of the cell comes in. So now you get this cross-bridge formation, but you get no contraction. So, that's why if the patient's in this position, when rigor sets in, it's like that's the position that the muscles basically lock up in. So muscle relaxation requires ATP, and ATP is not being made after death. Right? There's no aerobic respiration taking place. Now, ATP production causing this stiffening, they're going to be contracted until the filaments themselves start to decay. They're proteins, so they'll break down and decay after a period of time. So rigor peaks at about 12 hours, and then it starts to diminishing over the next 48 to 60 hours. So what I'm going to show you right now is I'm going to show you a video of the sliding filament model. It's got some really cool music. It's like Enya-ish.
We move our bodies by contracting skeletal muscle. While the results of this action are plain to see, the mechanism behind the action is not.
Okay.
Contraction of skeletal muscle
Sarcomere.
tissue occurs when actin and myosin myofilaments slide past one another, causing the sarcomere to shorten. It is important to note that the individual myofilaments do not change length themselves, but instead overlap each other to shorten the sarcomere. Moving in for a closer look, we can see the details of how this occurs. The molecules you see here are myosin, which at rest has both adenine diphosphate and phosphate molecules
See the two
attached
heads
to the
of
individual
myosin?
myosin heads: actin, tropomyosin, and troponin.
So actin's blue in this picture instead of
When we
red.
decide to move, calcium is released from the sarcoplasmic reticulum. These molecules bind with the troponin molecules. This causes the tropomyosin molecules to move, revealing myosin attachment sites on the active myofilaments. The single phosphate attached to the myosin heads gets released as the heads move to connect with the attachment sites on the actin. The resulting formation is called a cross bridge. The remaining adenine diphosphate molecule is then expended in what is called a power stroke, when the myosin heads pull the actin inward. Once the adenine diphosphate attachment is spent, adenine triphosphate molecules attach themselves to the myosin heads. These molecules trigger the release of the myosin heads from the actin attachment sites and are immediately broken down into adenine diphosphate and a single phosphate.
So now it's
as the heads
recocked
move
and
back
ready
into
to bind
resting
again, right?
position. This is called the recovery stroke. If calcium is still present, the cycle will repeat again,
So
and
here's
the
calcium.
sarcomere will continue to shorten until the calcium ions are transported back into
You
the
see how much
sarcoplasmic
ATP is
reticulum
being
and
used?
the muscle relaxes.
If you want me to rip the soundtrack, I will. So yeah. Okay, so what do you think? Is that helpful? To see it kind of in motion? See it in motion, I think it actually is pretty nice to be able to appreciate how it works. Okay, so as we wrap up today, I got a couple of possible test questions for you. Okay, yes, ma'am.
So when it re-costs the energy that's from the ATP from the action, or is it ATP coming off that?
So in order for when it's after it's fired, right, and it's still bound, it needs ATP to bind to let go. And as soon as it let go, it recocks. And when it does this same motion, ATP binds, it let go, and when it recocks, it goes to ADP. Does that make sense? So one cycle, right? So one cross bridge, power stroke, release. That's all one ATP molecule. So remember, every time you cleave a phosphate bond, you release energy. So adenosine triphosphate has basically 3 forms of energy. So ATP will go to ADP with PI, with a free liberated phosphate. And then ADP can go to A&P, adenosine monophosphate. So there's 3 different energy levels of ATP. So one full cycle burns 1 molecule of ATP. Okay? All right, so what surrounds each muscle fasciculus and what surrounds each muscle fiber?
It is B.
B, perimysium and endomysium. Very good. Peri is like the perimeter, endo is inside. The enlarged areas of the sarcoplasmic reticulum near the T-tubules are called what? Sarcolemma, sarcomeres, terminal cisternae, troponin molecules, or tropomyosin molecules. C, terminal cisternae. Very nice. Any questions? 3:32. Yeah. I'll see you on Thursday.